We start again. Okay. And now we give some examples. When studying molecules that are involved in biological molecules and compounds, there are some chemical groups that have typical signatures. For example, the carbonyl group that is present in the aldehydes, in the ketones, has got a typical absorption band in the visible. The peptidic bond that is formed when you bind two amino acids, it has a typical signature. Conjugated double bonds in organic molecules, they have got, for example, linear polymers like carotenoids, retinol, and in cyclic polymers like in porphyrins, hemoglobin, phthalocyanins, or whatever, or in aromatic amino acids or the basis of nucleic acids, these have typical signatures. Ions in transition metals, they get typical signatures like the, for example, iron in hemoglobin. Now, let's give some examples. For example, chlorophylls and carotenoids. This is the chemical structure of chlorophyll, and it is really, 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 really very much similar to hemoglobin. And instead of iron atom in the middle, you have a magnesium atom. And you know that this is the base of a photosynthesis process. Carotenoids that are present in plants, these are linear polymers, and they have got some chemical groups at two extrema, but these are formed by a conjugated backbone where you have alternance of single and double bonds. Okay, so you have conjugation of charge along this direction. Now, if you watch the absorption spectrum of this type of molecules, you discover that, for example, chlorophyll A, it has got an absorption spectrum like this. So this is peaked at about 430 nanometers, this peak here, the solid curve here. Then there is a minimum, there is no absorption here, in between 500 and 600 nanometers. And then there is a strong absorption peak at 680 nanometers, and then it goes down. So there are two main absorption bands. And as I said you before, you do not see any particular structure. These are bands that are due to the superposition of many transition levels, okay? But for example, if you look at the absorption band of carotene, this absorption band is peaked here around 500 nanometers. Okay. Now, always keep in mind that 430 is violet light. Okay. 480 is blue. 520, 540 is green, more or less. Red, 600 is orange. 630 is about red, and this is dark red, and then you have the infrared region. And these molecules do not absorb in the infrared. Now, when you are in summertime, okay, the leaves of the trees look like green, okay? Because in the leaves there is much chlorophyll, okay? And these leaves absorb light from the sun, and the energy from the photons of the sun is used to carry out the photosynthesis, okay? In this period here, what has happened during summer? It has happened that the chlorophyll has absorbed light and transformed in energy, and then the chlorophyll molecules starts from the ground state, goes to an excited state, and the extra energy is used to produce sugar, okay? And then the molecule goes back to the ground level, okay? And then another photon arrives, and another cycle, and another cycle, and another cycle, and another cycle. And these molecules cycle all over summer, okay? But there is a certain probability that when the molecule is in the excited state, it reacts with oxygen, it reacts with something else, it breaks, it is damaged, and then the chlorophyll is destroyed. That is the why during summer the sun is burning the chlorophyll molecules, and the leaves turn from green to yellow and to red. Okay? And why do they turn to a different color? Because only the carotenoids remain in the molecule, and the carotenoids have a different absorption spectrum, and so they look like a different color. Okay? Now, here, with the green curve here, that is shown here, this is, if you, in quantum mechanics, I believe you discussed the black body radiation. This is the black body radiation of a body that is at 5,777 Kelvin. This is the effective temperature of the corona of the sun. You know that light in the sun is not emitted by the bulk of the sun, but it is emitted by a very thin layer that is around the sun, where there is a recombination of atoms. And the temperature of this emitting layer is about 5,770 Kelvin. And if you calculate the black body radiation, you will see that the spectrum, the emission spectrum of the sun, and here you have the wavelength in a logarithmic scale. Okay, so this is 0.1 microns, this is 1 microns wavelength, and this is 10 microns. So don't make mistakes. The visible range is here, is below 1 micron. You remember, it is below 700 nanometers and above 400. So the visible range is here. Okay, this is the infrared range. So the light that is emitted by a black body like the sun in the infrared is it's not much. Okay? And you see that the absorption is peaked at 500 nanometers. So the black body radiation of light, and the green curve has as an axis here the intensity. So here you have the intensity of light measured in watt per square meter. Okay, and the intensity is as large as this. Okay, and the peak is at 500 nanometers in the green. Light from the sun is peaked in the green. Okay, the largest content in the green. Now, what would you expect concerning chlorophyll? Where should chlorophyll absorb light? To create an efficient leaf that is absorbing light, where should chlorophyll be have its absorption maximum. If the sun is peaked at 500, where should it be? The maximum absorption of chlorophyll. No, no reply. If you want to capture the sunlight, the energy from the sun, you want to absorb the photons. If most of light is at 500 nanometers, where do you have to capture light? At 500 nanometers. Where should it be the maximum of absorption of chlorophyll? It should be at 500 nanometers. But look here, there is a hole here. The chlorophyll has an absorption spectrum with a hole at 500 nanometers. And then I ask you this question. If the intensity of the sun is maximum in the green, why the leaves of the vegetables are green? What is happening when light from the sun is hitting a leaf? The frequencies that are absorbed, and the frequencies that are not absorbed are reflected. Now, if the leaves are green, it means that the green is reflected, so it is not absorbed. And now my question is, if the sun is peaked in the green, why the leaves do not take the green? If you ask this question to most of professors working in photovoltaics, they cannot reply. Why? Now, this is the why here. I plot the black body spectrum, but referred to the right-hand axis. So this is number of photons per square meter. Okay? Number of photons per square meter. And I need to use the blackboard. Just one. The whiteboard. Okay. Think you have a pen. Here it is. Think you have light. Okay? With a certain intensity. Okay? Classically, the intensity, we said, is the square modulus of the field. Okay? Classically. But in quantum mechanics, we said that what is the intensity in quantum mechanics? Is the number of photons per second times the energy of the photons. Okay? These are two descriptions of the intensity. Okay? Now, there are some prefactors here. No? But let's say that we think that these prefactors are one for the sake of simplicity. Now, look at this expression here. It means that this is average intensity. This is average number of photons. Okay? Look at here. You can say that the average number of photons is equal to the average intensity divided by the energy of a photon. Okay? What does it mean? That if you are at low frequency, so if you have light at low frequency, for example, a radio wave, okay, the energy that each photon is bringing is small, okay? So for a given intensity, the number of radio wave photons is very large, okay? Large average number. Okay? If you are at high frequency, for example, gamma rays, then the energy of a photon is very large, and for a given intensity, the average number of photons is small. Okay? Small. Average number of photons. That means that depending on the frequency, a given intensity, a given classical intensity measured in watt per square meter correspond to a different flux of photons. Okay? So if you have 1 watt per square centimeter intensity, this may be million of photons in the radio regime and 1 photon in the gamma ray regime. Is it clear? So the average number of photons is the average intensity divided h bar n, but the frequency is connected to the wavelength. We know that, remember, wavelength is in vacuum the propagation speed divided by the frequency. So you have that the frequency is c over lambda, and here this is I average divided h bar is c times lambda. Okay? So the average number of photons depends on the average intensity and on the wavelength of light. So here in the slides, if you look at the black body curve, the green curve is the classical intensity, and the red curve is the fluence, number of photons per unit time per square meter. So how many photons arrive to you? And this curve is distorted because when you calculate the average number of photons, there is a lambda inside the formula. And where is the peak? At 600 photons, 640 nanometers. So in the radiation coming from the light, the intensity is peaked at 500 nanometers, but the average number of photons is peaked at 640 nanometers. So the maximum fluence is in the red. Now look at this curve that you have here. Where is the absorption of, sorry, I touched. What did I do? Where is it maximum, the absorbance of the chlorophylls? Around 640, 650 nanometers. And why is it maximum there? Because the absorption process is a quantum process. A molecule must absorb a photon. So it is not important the classical intensity. It is important the number of photons you get per minute, per second. Okay? So the chlorophyll is optimized to match the fluence of the sun and not the intensity. That is the why the leaves rejects the green, because in the green, at 500 nanometers, the content, the number of photons per unit time that gets to the leaf is smaller, because the photons in the green, they are a bit more fat, they are larger, and so you have less photons per unit time. That demonstrates why that the absorption process is a quantum mechanical process. Now, let's look at the amino acids. If you take some amino acids, did you have the exam of biochemistry with Pagnanelli? did you measure, you have not yet measured the absorption spectrum of tyrosine, tryptophan. The amino acids show typical absorption bands. You see here, this is the wavelength, this is molar absorptivity, that is the molar extinction coefficient that the epsilon that we described above. Here we are in a logarithmic scale, and you see that tryptophan absorbs in the ultraviolet at about 280, tyrosine a bit smaller wavelength, phenylalanine at a smaller wavelength, and they have a typical shape. So if you measure the absorption of a solution, you can sometime understand which amino acid you have inside the solution. Okay? And given that amino acids are in proteins, you can understand which proteins you have in front of you. And these are what we call endogenous chromophores. These are absorbing molecules that you naturally have in living tissues. Amino acids, chlorophylls, carbonyl groups, peptidic groups, these are naturally. But if you work in biology and you want to use a microscope, you can use exogenous chromophores. What is a chromophore? It's a molecule that is colored. It brings color. Okay? And there are several different molecules that are used to stain the biological tissues and perform microscopy. Now, you have to keep in mind that starting from the Egyptians, okay, to the Greeks, to all the people that have been living in the world, one of the most important industries that we have ever had is the industry of tissues that you use to dress. Okay? And people learned to change the color of wool and cotton by using any type of coloring agent. So when people started to use chromophore in microscopy, the first thing that happened was that the chemists opened their drawers and they took out of the drawer, oh, look, this is red, Texas red. This is used to make nice t-shirts from fruit of the loom, okay? This is DAPI. This is malachite green from the ancient Egyptian tradition. So many of the dyes that are used to stain tissues, they come from the tissue industry. Of course, you have to select dyes that are not poisonous for living tissues. But it is almost every time true for dyes used in tissue industry, because if you wear a shirt, okay, that is stained with a dye that is poisonous, after some time, you get bubbles, you begin scratching, you feel sick, and you have to remove this. So they are already compatible somehow. Okay? And here you have the first three molecules that have been used to stain tissues. They were the most diffused until 15 years ago. They are named fluorescein, DAPI, and Texas red. In the laboratory work, in the next laboratory work, we will work with a molecule that is very similar to this Texas red that is named rhodamine 6G. It's the same family. Okay? And you see that they have got a certain structure. There are some atoms involved in the structure of the molecules. And here you get the absorption, the typical absorption band of these three different molecules. And you see, and you will understand when we will discuss fluorescence microscopy, that the absorption bands of these three molecules, they are well separated from each other. That is, DAPI absorbs with an absorption peak at about 350 nanometers. Fitch or fluorescein absorbs with a peak at about 480 nanometers, and Texas red absorbs with a peak a bit less than 600 nanometers. And the three absorption bands are well separated. Now you see that Texas red has got a main peak and a vibronic replica here. Okay, so this is another transition to a highest excited level. And be careful, because here you get wavelength. So smaller wavelength means larger energy. You remember the figure that I've shown here before? Okay, I was plotting as a function of the photon energy, and the photon energy is inverse to wavelength. Okay, so you see that you can see some, but here the replica is a shoulder. You cannot see it, and here there is something, but it is outside the spectrum. Okay, so you can, most of these molecules, they are routinely used in microscopy. And these bands come out from electronic transitions between a ground electronic state and a first electronic excited state, and possibly with some vibrational contribution. Okay? Here you have the example of the cyanins. These are the most diffused dyes used in microscopies at the moment. So you can buy them on a catalog. You log in on the Thermo Fisher website. On Thermo Fisher, you can buy the SARS-CoV-2 virus, the spike protein, the N protein, the antibodies against the spike protein. You can buy the dyes. You can buy anything you need in biology. Okay, and these cyanine, how are they done? Their chemical structure is based on two indole groups, two external groups that are joined by a conjugated bridge with double bond and single bond. Okay, a molecule like this shows an absorption spectrum, for example, the blue line here, so this absorption spectrum here, that is peaked a bit below 550 nanometers, and then it is like that. And there is a vibronic replica here, okay? If you change the length of this bridge, and for example, you change by half single double bond length, and you go to the 3.5, then you see that the absorption spectrum is shifted towards the red, towards the longer wavelengths. So simply by adding a double bond here, you will change the spectrum like that. If you go to cyanine 5, that is 5 repetition units here, you see that the spectrum gets the green curve here. And if you go to cyanine 5.5, you go to the yellow spectrum here. So this is a class of molecules that can be tuned to any wavelength by simply changing the length of the conjugated chain in between. Okay? Now, we will go back to these properties of the cyanins when we will discuss fluorescence. For the moment, you have a list of reasons why the cyanins are better than the standard traditional dyes. And there are some reasons. The absorption and emission spectra are similar to those of the traditional dyes. They have better water solubility, so they're better soluble. They are less sensitive to pH and to the organic environment. They are more photostable, and they have a large quantum yield. But these two last things we didn't introduce yet, so we don't discuss. You have another family of dyes that are the so-called Alexa fluor dyes. And these are names given by companies. This dye here is from the Höchst company in Germany, okay? And these are based on the rhodamine 6G also structure, And depending on the side derivatives that you attach to these structures, you get different absorption spectra. Now, there is a very nice software that is named the Spectra Viewer, and now I will show to you that you find on the internet, and you can see the absorption spectrum of these molecules. Let's see this. Let me Google this. Now, first of all, I show you, I share my screen. Okay, I Google. How can I Google? Okay, here, Spectra Viewer from Thermo Fisher. And in this Spectra Viewer, you can choose a particular fluorophore, cyanine, and it gives you a list. APC, PE, cyanine 7, cyanine 5.5. Let's take cyanine 5, and here you get the absorption spectrum. Let me. No filters, no plot normalization. And this is the absorption spectrum of this cyanine 5. So you see that the absorption is large here, then there is a second peak, and then it grows. And here, this is not a peak. This is just the end of the region where you measured the absorption. Okay? But instead of this molecule, you can take another molecule, for example, rhodamine 6G, that is the molecule that we shall use in our experiments. And you see that this is the absorption band of rhodamine 6G. This is peaked at 527, and there is a shoulder here and maybe another shoulder here. Okay? And on the same catalog, you find antibody. Let me see. The Alexa fluors, for example. This is the Alexa fluor 430. So if you go on a Spectra Viewer, you will find all the absorption spectra of all dyes used in applications. Okay? So let's go back to this. And with this, we stop. We end with the lectures, group of lectures number 2. So what do you expect? That when you have organic molecules and you take them, you put them in solution in a solvent, but the solvent could be the water that you have got inside living tissues. If you put these molecules inside a cell, they will dissolve in the water that is constituting the cell. When these molecules are in solutions, they show typical absorption bands that come out from all the processes that we discussed. Okay? Now, it's 17:53. We take 10 minutes rest, and then we pass to the group of slides number 3 that I forgot to send you in advance, but I will send you after the lecture. So we stop for 10 minutes.